dear students. Today I will discuss on immunoglobulins, types of immunoglobulins and their functions. We know that immunoglobulins are soluble protein molecules which are involved in immune system, and these molecules, these proteins are secreted from B lymphocytes. B lymphocytes are generated in our bone marrow, then they move in our blood and lymphatic system. And on the surface of B lymphocytes, there are two types of immunoglobulins present: IgM immunoglobulin and IgM and IgD. Now, when a B lymphocyte encounters an antigen, the B lymphocyte gets activated, it is converted to plasma cells and memory cells. Plasma cells immediately secrete IgM isotypes, and memory cells keep the memory of that particular antigen, and when the same antigen again enters into the system during the second response to the same antigen, the memory cells quickly convert it to plasma cells. This time the secreted immunoglobulin can be IgE or IgD or IgA. So there are 5 different types of immunoglobulins present in our serum. Now, each immunoglobulin molecule is composed of 2 heavy chains and 2 light chains. Each light chain is joined to each heavy chain through the interchain disulfide linkage, and 2 heavy chains again joined together by interchain disulfide linkages. Thus, these 4 polypeptides, 2 heavy and 2 light chains, they form the complete immunoglobulin molecule. And each light chain has a molecular weight of 25 kilodalton, and each heavy chain has a molecular weight of 50 kilodalton. Thus, an immunoglobulin molecule has a molecular weight of 150 kilodalton. Now, from the N-terminal end, 100 or 110 amino acid residue is called variable region, because the sequence of this region varies among different immunoglobulin molecules, and this region is responsible for antigen binding. And if we look into the detailed structure of the variable region, then we will see 3 discrete regions on the light chain and 3 discrete regions on the heavy chain variable region are hypervariable in nature, and these 6 hypervariable regions are responsible for binding to an antigen molecule. So these 6 hypervariable regions are called CDRs, that is complementarity determining region, and rest of the heavy chain and rest of the light chain is called the constant region, and the constant region is responsible for the biological activity of an immunoglobulin molecule. And both the heavy chains and light chains, they contain 110 residue long homologous regions called a domain or a unit. The light chain contains 2 such domains, one at the variable region, one at the constant region, and the heavy chain contains either 4 or 5 homologous regions. Now, the sequence of the constant region of heavy chain can be 5 different sequences have been found for the constant region of heavy chain. When the sequence, we named the sequences differently. If the sequence is gamma, then we call that isotype IgG. If the sequence is mu, we call it IgM. If the sequence is alpha, we call it IgA. If the sequence is epsilon, then we call it IgE. And when the constant region sequence is delta, the immunoglobulin is called IgD. Now, for the light chain constant region, 2 sequences are present in our system, either kappa or lambda. So, the molecular formula of IgG will be either gamma 2 kappa 2 or gamma 2 lambda 2. Clear? Similarly, for IgE, the molecular formula will be epsilon 2 kappa 2 or epsilon 2 lambda 2. Now, IgG is the most prominent or major isotype found in our blood. IgA is present at 10 to 15% in our blood, and IgM is approximately 10% in our blood. Now, I will tell you IgM. IgM is the membrane bound immunoglobulin. It is present on the surface of B lymphocyte. Clear? and it accounts for 5 to 10% of total immunoglobulins in serum. Now, as soon as the B cell gets activated and it secretes IgM, the IgM becomes pentameric. Now, in pentameric IgM, 5 monomeric units are joined together by disulfide linkages, interchain disulfide linkages between the CH3-CH3 domain and CH4-CH4 domains. And we can see an extra polypeptide called J chain in the pentameric IgM. It is bound to 2 CH4 domains of 2 mu chains of 10 mu chains present in the pentameric IgM, and J chain is responsible for polymerization of IgM monomers, and it joins the pentameric IgM just before secretion. Now, polymeric IgM, it can bind to 10 small antigens, or if the antigen is larger, then 5 or fewer number of antigens can be bound to the antigen binding sites of a pentameric IgM. That means it has a higher valency compared to the other immunoglobulins. Now, since IgM has multimeric antigen binding sites, it is very useful in the neutralization of virus or bacterium, and it has been found that RBCs, when RBCs bind to specific antibodies, they precipitate out, and this process is called agglutination. It has been found that it takes 100 to 1000 more IgG molecules, okay, over IgM to produce same level of agglutination. Okay. Similarly, less IgM is required to neutralize virus or bacterium. And IgM is very useful in activating complement system. Complement system is a part of our immune system. It is composed of a number of soluble proteins which are present in our serum as inactive molecules. It is activated when antibody binds to an antigen. Classical pathway starts with antigen-antibody binding, and to initiate the classical pathway, at least 2 Fc regions of an antibody must be present in close proximity. It is achieved with only 1 molecule of IgM, whereas 100 of IgG molecules are required to activate the classical pathway of complement system. So, IgM is more advantageous in activating complement pathway. Now, I will tell you about IgD, which is another membrane-bound immunoglobulin on the naive B lymphocyte, which has come out from the bone marrow into blood. It contains both IgM and IgD. But no biological function, effector function has been found so far for IgD. Now, I will tell you about the other isotypes which are secreted from the memory B cells. Okay, that means during the second exposure to the same antigen. Now, what happens? Some small molecules called cytokines or interleukins are secreted by many of the immune system cells.
Ma'am, I have a question. What are interleukins?
Interleukins are small signaling protein molecules which are secreted from lymphocytes, and they act on other lymphocytes. So, in response to different interleukins, the memory B cells in the presence of the same antigen are converted to plasma cells. Normally, on the memory B cell, the constant region of the heavy chain is of mu isotype, but in presence of some interleukins, the mu is changed to either G or alpha or epsilon. Clear? Like in presence of interferon gamma, the mu isotype is changed to gamma constant region. Thus, now the plasma cells secrete IgG. Or in presence of IL-2 or IL-4 or IL-5, mu remains the same. Now the plasma cell secretes IgM only. However, in presence of TGF-beta, okay, TGF-beta influences the memory cell in such a way that now the mu constant region heavy chain is changed to alpha. Clear? Now IgA is secreted. So, depending on the cytokines secreted by T cells, T lymphocytes or macrophages, the memory cells change the constant region of the immunoglobulin molecule, and now the other isotypes are secreted. This is called class switching, since the isotype class is switched from one isotype, mainly from IgM to other isotypes. This is called class switching, and this is very important since the IgG, IgE, IgA, they have specific functions. Now I will tell you about IgG, the major component of immunoglobulins. It accounts for almost 80% of the total immunoglobulins present in the serum, and it is produced during the second exposure to the same antigen, where the mu constant region is changed to gamma, but the antigenic specificity remains the same. Now IgG can be divided into 4 subtypes or sub-isotypes. IgG 1, 2, 3, 4, and these proteins, these 4 proteins, they are encoded by different germline genes, and the DNA sequences are 90 to 95% homologous. Now if we look into the structure, then structurally they differ in the length of the hinge region and also in the number and position of the disulfide linkages, and they are named 1, 2, 3, 4 depending on the decreasing serum concentration. Percentage of IgG1 is maximum in serum and percentage of IgG4 is least in the serum. Now IgG plays a major role in immune system, like IgG1, 3, 4 can readily cross the placenta and protect the fetus. Now IgG3 is the most effective complement activator, and IgG1 and 3 can act as opsonins. Opsonins means they enhance the phagocytosis of the antigen. These molecules IgG1, 3, they cover the antigen, and there are receptors on the phagocytic cells against the Fc region of antibodies. So the antigen covered with antibody is very easily phagocytosed by the phagocytic cells. Thus they help or enhance the phagocytosis of the foreign molecules. Now I will tell you about IgE, another isotype present in our serum, but it is present at a very low concentration in our serum. And IgE, it is mainly secreted in case of parasitic infection, but it is secreted in atopic individuals. In these individuals, normal environmental molecules act as immunogens, which are otherwise non-immunogenic. Okay, and these molecules are called allergens. Okay, and the state is called atopy, and the individuals are called atopic individuals. So what happens, suppose a normal non-immunogenic molecule comes and it binds to a B cell receptor, to the IgM present on a B cell. Now in those individuals, atopic individuals, in response to that normal molecule, the interleukins secreted, they help to activate the memory cells, or in presence of those cytokines, IgE isotypes are generated. Now once the IgE isotype is generated, these molecules will bind to the mast cell and basophil surface. Mast cells and basophils have receptors for the Fc region of IgE molecules. So the IgE released will bind to the receptors present on the basophil and mast cell surfaces. These cells are sensitized. Next time the same antigen enters into the system, they will bind to the sensitized mast cell basophil, and binding of the allergen to the IgE will trigger the mast cell and basophil to release many molecules present in them. And these molecules are mainly responsible for causing vasodilation, vasoconstriction and many other changes in the physiology. So, it leads to a kind of hypersensitivity, type 1 hypersensitivity. This is also called allergy. So, IgE is mainly responsible in allergy. Now, I will tell you about IgA. IgA can be found as in its monomeric form, or it can be found as a dimer or trimer or a tetramer. The two in its dimeric form, two IgA molecules are joined together by a J chain, just like pentameric IgM. The J chain is bound to the terminal CH3 domains via cysteine interchain disulfide linkages. Now, polymeric IgA is present in different secretions like tears, saliva, mucous membrane of digestive tract, respiratory tract, genitourinary tract, in all different secretions, also in breast milk, etc. And a human secretes almost 5 to 15 grams of secretory immunoglobulin A per day. And the mucous membranes of our system are full of IgA secreting plasma cells and memory cells. Since the secretory IgA is present in the secretions, that is in different portals of entry, okay, they prevent the entry of foreign pathogens. They, as soon as the foreign pathogens enter into our system, these secretory IgA molecules cover them, bind them and prevent their attachment to our mucosal cells. Thus, they prevent the infection. Also, IgA present in the breast milk provides protection to the newborn baby because immunity develops after several months. For the first month of life, almost no immunity is present in a newborn baby. So, the IgA coming through the breast milk protects the baby from its environment. Now, I will tell you how the polymeric immunoglobulins are secreted into the secretions. So far, we have studied two polymeric immunoglobulins. One is IgM and the second one is IgA. IgM is always found in pentameric form, but IgA can be dimeric, trimeric or tetrameric. Now, how they are secreted in the secretions? There are receptors called poly-Ig receptors present at the basolateral surface of epithelial layer of genitourinary tract, respiratory tract, etc. And also poly-Ig receptors present in the glandular epithelia, like in the mammary gland, lacrimal gland, etc. Now, what happens? The poly-Ig receptor binds to the polymeric immunoglobulin. And as soon as the polymeric immunoglobulin binds to a receptor, it is endocytosed and the vesicle lumen side of the epithelium layer. As it fuses with the lumen side apical surface of the epithelial cell, the molecule is exposed. Now, the enzymes present in our digestive tract or other tracts, they will cleave the molecule. So, now the poly-Ig receptors remains bound to the secreted immunoglobulin and it is called a secretory component now. It looks like this, and presence of secretory component helps how it covers the protease susceptible area so that the immunoglobulin remains for long in our secretions. And IgA is a major secretory immunoglobulin. IgM is found in less amount because of its large size. It can't diffuse well, but you can find a small amount of IgM in all secretions. But however, IgA is the major component of secretory immunoglobulins. Now, since immunogens are glycoproteins, they can also act as antigens when injected into another animal species. And three types of determinants or epitopes are found on an immunoglobulin molecule, like isotypic determinant, allotypic determinant and idiotypic determinant. Within a species, all the members express same isotypes. But different species, in different species, the isotypes differ because the genes are different. So, if an immunoglobulin is taken from one species and injected into another species, then some parts of the constant region domains will be considered as foreign and antibodies will be developed. These determinants present on the constant region are called the isotypic determinants. Like rabbit anti-mouse IgG. Suppose if we inject mouse IgG into rabbit, then some of the epitopes present in the constant region heavy chain of mouse IgG will be considered as foreign and antibodies will be generated against it. This antibody is called rabbit anti-mouse IgG. It is against the isotypic determinant. And these antibodies, anti-isotype antibodies, are very useful in research as well as in diagnostic purposes. Next is allotypic determinant. Like I told you, IgG can be of 4 sub-isotypes, IgG 1, 2, 3, 4. So, if we take the suppose IgG1 from one member of a species and inject it into another member, okay, having different allotype, then the determinants present in the constant region heavy chain will be considered as foreign and antibodies will be developed. These determinants are called allotypic determinants and anti-allotypic antibodies are generated against it. Like allotypic determinants are generated during pregnancy in the mother in response to paternal alleles present in the fetus. And another determinant is called idiotypic determinant. Suppose we have an immunoglobulin raised against antigen A. Now, if we inject this immunoglobulin into a recipient identical with the donor, okay, then all the constant regions are same, but the recipient has no immunoglobulin against antigen A. Now, its immune system will recognize the variable regions as foreign and antibodies will be developed against the variable region determinants. These determinants present on the variable regions are called idiotypic determinants and anti-idiotypic antibodies will be developed against it.